Okay, so our last talk of the session is from Clément Bernard from University Paris-Saclay. He's going to be talking to us today about recent advances, and limitations of RNA 3D structure prediction.
So, hello everyone, I'm Clément Bernard and I'm a third-year PhD student. And today I'm going to talk about recent advances and limitation of RNA 3D structure prediction. My thesis is supervised by Farid Zataï and Guillaume Postic, which are from Ibisque in France, in Évry, and also by Sarah Ganet, which is from the Lycée, which is also near Paris. Maybe a little explanation about the context where RNA molecules are like molecules involved in multiple biological processes, like diseases. And we know that the biological function are linked to its 3D structure. So there is an interest of just knowing the 3D structure from the sequence. And so it has been a challenge for years. And so this talk is really like a state of the art of what exists in terms of RNA 3D structure prediction. So at first, we can have a description of RNA with just the raw sequence, with just a linear combination of nucleotides. And then we could have details about some complementary base pairing, which would be like considered as a secondary structure. And then we'll have the complete folding of spatial arrangement of the atoms, which will be the 3D structure. And we know that the 3D structure is constrained by its secondary structure, which is also constrained by its raw sequence. And so what I will be talking about today is really given the PDB data that we have, what are the kind of models that can just predict the structure given like all the data that we have. So there are three main types of approaches. The first one, which was also discussed in the previous talks, would be the ab initio. Then we would have the template-based. And finally, more and more methods try to take benefit of the data in the PDB, which will be like the deep learning method. But at first, what could be the features that are used to predict the RNA 3D structure? So we could have just the raw sequence, whereas the advantage is that it's just a simple to use, just sequence of nucleotides, and it will give the different type of nucleotide that we have. But it is limited that we do not have any more interaction that are encoded as the inputs. Then some methods will try to use the secondary structure, which will help guide the prediction, as there are some complementary information inside. But it is also limited in the fact that for long sequences, it may be hard to have the secondary structure, and it also will depend on other predictive models, which might be, yes, a limit. There are some methods that try to start with just a tertiary structure. So it could enable to have a good start for the simulation, but the drawback would be to require to have a prediction, which is not always the case. And finally, as for proteins, we could use the multiple sequence alignment as input. So it could give information about the alignment with known families, but it will require to have and to know those kind of families, which is not always the case for RNA. And so it might be hardly generalizable for RNA with unknown families. So this is one of the drawbacks for the MSA. But what about the different type of approaches that exist? So for the ab initio, the goal is to, so they rely on the fact that native structure are those with the lowest energy for RNA. And so they will try to reproduce the physics of a system with some energy based on dihedral angles, bond lengths, etc. And they will use some generation process to try to have some candidates, RNA candidates, and then they will try to discriminate, to rank those candidates given a scoring function, for instance. And the sampling procedure we usually are usually Monte Carlo or molecular dynamics, for instance. And in the generation process, they will try to add some small changes in the structures, and then to see if it will add a benefit to the energy of the molecule. And for those ab initio method, but even for those methods, we can have a coarse grain representation. So instead of having for one nucleotide, for one residue, like all the atoms, we could just consider some small amount of atoms per nucleotides. And so it will be a coarse grain representation. And the goal is really to reduce the computation cost, but it has also its drawbacks. So it will lose the quality of the representation. And at the end, it will require to have a step to go from the coarse-grained representation into the full atom structure. And so each method will have a different coarse-grained representation depending on their different solutions. And at the end, to have a final structure, they will have like a lot of candidates. So they will compare them in terms of root mean square deviation, and then try to cluster them to have the more representative structures. And then they will fix the broken bonds and steric clashes, for instance. But for this ab initio method, the main bottleneck will be the sequence because it's really slow. We could have multiple hours for just one prediction, and whenever we increase the sequence, we'll have really increased the size of the sampling molecules. And so it also requires to have an efficient sampling method to avoid useless computation. And then we have also the template-based approaches. So this kind of approaches will, the idea will be to create a mapping between some non-templates. So given some fragments, which could be just 3 nucleotides, for instance, or just secondary structure elements, they will create like a template database with this mapping. And then for the inference part, they will try to align the different smallest secondary elements, for instance, to its 3D templates, and then try to assemble everything. So here is an example of a model which is called a 3D RNA. And I show it because it's kind of a good representation of the template-based approaches. So given a set of RNA, they will create SSE templates library. And then, so given a new sequence with the secondary structure, they will decompose it and then have a look in the template library to find the 3D fragment associated with the secondary elements. So we could have multiple. And then they will try to assemble it to have some candidates, and then try to choose between the best candidates with some ranking with their custom scoring function. And at the end, they will do some optimization, etc. So it's really the idea of the template-based approaches. And they will rely on representative templates. So this may be limited by the amount of data that we have for the RNA 3D structure. And so sometimes it can fail to generalize to ancient structure because there is no available templates. And finally, we have the deep learning method. So the deep learning method, in fact, it was with the success of AlphaFold 2 and the Nobel Prize for proteins, there have been work to try to adapt it to RNA. So at first, it was not possible to use AlphaFold 2 architecture for RNA due to the differences in terms of vocabulary, the disparity in length compared to proteins, and we also had fewer structures available for RNA. So there have been some work to try to adapt this architecture to RNA before the release of AlphaFold 3. And so to just understand what do we predict with a deep learning approach using kind of AlphaFold 2 architecture. So given two sets of points on the left side, for instance, we may want to apply a transformation, so rotation and translation to the green point to get as close as possible to the red one. And so we can do it with just rotation and translation. So here, just rotation matrix and translation vector. And if we apply it to the red point, we'll get as close as possible to the red one. And so the idea would be, instead of having just point, we would have some atoms for one residue. And so the goal would be to predict for each sequence rotation and translation matrix to convert from like a base frame into its global position in the structure. So here is really the idea of like the kind of deep learning approaches that will reproduce AlphaFold 2 architecture. So really the goal predicts the rotation and translation matrices for one nucleotide to get the coarse-grained representation in its global skeleton. And then to get the full atom reconstruction, they will predict the torsional angles that will convert from this coarse-grained representation into the full atom, which will depend on the frame representation and so the coarse-grained representation of each method. But still, the overall performances of the deep learning method are still need to be improved. And so it is limited by the available PDB data that we have. And there is also no interpretation of the model for the deep learning method. And so we did a complete review and a benchmark of this existing method, so ab initio, template-based, and deep learning method, where the goal was to compare the user available method, which I will talk about later on. And then there was a release of AlphaFold 3, which in fact works also for RNA. And so they changed a little bit the architecture, so the last module. So before it was a prediction of the rotation and translation plus the torsional angles, and now it's more a diffusion module to generate the full atom structure. But what about the evaluation? So how do we evaluate this kind of approaches, so the template-based, deep learning ones, etc. So for the RNA, there were, before CASP, there was a RNA puzzle initiative, which was a community initiative with some challenges, just like CASP for proteins. And at the end, they try to have some collaboration with the CASP community to create a CASP RNA for RNA. So there are two types of standard evaluation sets. But in fact, the result from either RNA puzzle or CASP RNA are results from previous years. So the methods are not always up to date. And even the predictions are also obtained through like huge optimization of multiple parameters, which is not always reproducible. This is why we decided to do our own benchmark using really user available method. And we also included AlphaFold 3 with just a default parameter to see at this moment what are the best methods. And so we did it using different benchmarks, so different datasets. So we used the RNA puzzle and CASP RNA datasets. We also created a set of independent RNA in terms of RNA family from RNA solo, where RNA solo is a work to have some clean data from the PDB. And there is also another work, which is RNA 3DB, where the goal is to create a set of RNA datasets, which are independent in terms of structural and sequence information. And so in these papers, they also talk about some orphan structures, which are advised to be used as a test set. So we use this one, which is the RNA3DB orphan database. And we also try to have a set with a long RNA, which we call RNA3DB long, which are mostly ribosomal RNA. And we ensure that the release dates of this RNA are like did not leak with the training set of the different approaches. And so here you have examples of the top two predictions, top rows two prediction for with AlphaFold in blue, AlphaFold 3, and in orange native structure. So we can see, for instance, for the RNA3DB long dataset, we have like long structures. And for RNA3DB orphan, for instance, we have like kind of different RNA. And we did the benchmark using different approaches. So we used 11 models. So for instance, we have 3 different ab initio approaches. So we already used the web server, with default parameters. We also benchmarked template-based approaches, and also some deep learning approaches, and also AlphaFold 3. And the challenge-based are just the best solution from different challenges, like for each challenge through the years. But what about the evaluation? So how can we evaluate the prediction of these different methods? So given native structure and predictive structures, native in orange and blue predictive structures, how can we assess the structural quality? So there are different types of approaches. There are 3 main types. So there are what we can call the general metrics, which will consider the differences for the general shape of the structure, like LRMSD, for instance. And then there are some more protein-based metrics, which are just taken by the protein community, like TM score, for instance. And then there are like RNA-oriented metrics, which will help to see the different limits for the different models, which will consider, for instance, RNA key interaction, or even torsional angles, alignment, etc. And so for the benchmark, we use a tool which we'll develop, which is called RNA Advisor, where the goal is really to gather in one command line like all the existing metrics and even some scoring functions. So we use this tool to have the results. So here are some of some interesting results that we got. So here we took like 10 metrics and we normalized for each method, each metrics to have the best values as 1 and the worst values as 0. And then we consider the cumulative sum of these normalized metrics. So the higher the better for each method. And so for instance, for RNA puzzles, we can see that this is AlphaFold 3 for RNA that outperforms the other method, which might be biased for the challenge based because it's all we have all method for this challenge based, for instance. For the Casper RNA, so as it was mentioned in the paper, this is not AlphaFold 3 the best method, but instead more human-guided method. And for instance, for RNA puzzle, this is SimRNA, which is an ab initio solution which has the worst performances, but which really might be due to the limit of ab initio method, which is not enough simulation time, as we use this as a web servers. And one interesting result also is for the 2 deep learning method instead of AlphaFold 3. We can see that they are for the MCQ and LCS10, which are like metrics related to torsional angles. They have some quite bad results compared to other methods, but they do have like a good global alignment, but they fail for really RNA specific features, which is not the case for AlphaFold 3, which has like really good results for this metrics, so for the torsional metrics, and yet they do not integrate any explicit geometry in the model. So it's an interesting result. And then there is the result for the RNA solo datasets. So we see that AlphaFold 3 has better results for this dataset, but for this one, we could not ensure any data leakage. And then there is the result, we have the result for the orphan structures. So we did just a prediction using one template-based, one deep learning, and one ab initio method, because we have like a lot of prediction to do using the web servers. And we can see that for this method, the results are not that good for the orphan structures. AlphaFold does a little bit better, but it still struggles for this dataset. And if we have a look at the top 2 worst predictions by AlphaFold 3, for instance, we can see that these RNA are in fact not single-threading RNA. There are more RNA that are inside a more complex molecule with some context that should impact its structures. And so we decided to predict the structure with the context for AlphaFold 3, because for the other method, we could not do it. So we do it for half of this dataset. So these are the results for half of the dataset with AlphaFold 3, but given the context. And we can see that it does better in terms of prediction for this dataset. But still, here we have, so the same in blue, AlphaFold 3 without any context. In orange, we have the native structures. And in purple, we have AlphaFold 3 with the context. And still, there are some cases where AlphaFold 3 need to be better for those orphan RNA, which is not an easy problem. And then we have for the RNA3DB, the long dataset with a ribosomal RNA. So it has some good performances. And we could only ensure this prediction for AlphaFold 3 because the other one would not predict for long RNA. But it also might be biased by the quality of data for ribosomal data in the PDB, which might be too similar and then lead to really good predictions. So for instance, the top 2 worst prediction has a TM score for 0.74, which is quite high. So it might be explained by some high similarity in ribosomal data. And so as a conclusion, so there are 3 main types of approaches for RNA 3D structure prediction. There are the ab initio method, which try to reproduce the physics of the model, but will require a huge simulation time. There are the template-based approaches, which try to create a mapping between structural elements, but they will struggle to unseen structures. There are also the deep learning method, which will lead to good general shape, but usually fails to reproduce RNA key specificities. And then we have also AlphaFold 3, which is a mix of deep learning method but that solves some current deep learning limits. And so we did a benchmark using 5 different data sets to compare what exists. And for some limitation that we found for AlphaFold 3, for instance, it's a need of context for orphan RNA. And there is still improvement to be made for RNA without homologue, which is the case for some RNA in Casp-RNA, in the Casp-RNA competition. And so for the future of the field, we still have improvement to be made to have the success of AlphaFold 2 for RNA. But the main bottleneck remains the lack of data that we have for RNA. Maybe also future direction for the field could be to try to predict the dynamics of the folding. And for instance, what I've been working on is to leverage language models to predict RNA structural features. So for instance, I've done a work to predict from the sequence the torsional angles. And so the idea is, as we are lacking of data for RNA 3D structure, we can leverage some language models which are pre-trained on just the sequence, and then infer some structural features from these pre-trained models. And just to let you know that all of our teamwork like is gathered in the Every RNA platform. So for instance, this talk is a mix between like a work on AlphaFold 3 for RNA and also state of the RNA art. So thank you for your attention.
Super, thank you for that lovely talk. Do we have any questions? I guess I can ask a question. What did I write there? Oh, yes. So I think maybe in the last talk there was Oleg made the point that things like PLDDT scores and these confidence scores are very, very important to judge the quality of your models. Is this something that has made its way into the RNA modeling deep learning methods as a whole yet? Or is this?
So usually they will try to do the same thing as AlphaFold 2. So there is a confidence head that will try to predict the LDDT and TM score. So it's like it is used for some method in the deep learning ones, but it's more like a scoring function for the other one for the ab initio and template-based approaches. But for the deep learning ones, yes, it's kind of the same confidence head to try to predict TM score LDDT.
Hi, Frederik, Aarhus University. Maybe a little bit out of at least the bioinformatics you talked about, but you mentioned that the orphan RNAs needed context in case of more correct folding. Do you see this as a big problem that a lot of the high quality RNA structures are co-crystallized with protein score? I know for EM it's also difficult to get atomic resolutions. Do you think that is a major drawback and that maybe MD would be better for gaining this model or this ability to predict simulation?
I don't know if I get the question, but I would say, yes, I think we may need to have more context to get better insight for the RNA structure, but we are already, the main bottleneck is what we have in the PDB. So if there is no context in the PDB, we could not get any context in the models. So usually some of the models that I presented are just trying to get this from the sequence, the outputs. And yes, indeed, with a lack of more context, it makes sense that it might fail.
Okay, do we have any more questions? Okay, in that case, I'd like to thank Clément once again.